Good morning, and thank you for attending Neuroscience Week. Today, we will hear from Erin Powery and Dr. Michael Bienkowski. Drs. Powery and Bienkowski will focus on specifically the VS200 slide scanner and research using whole slide imaging. Please stay tuned after the talk for a live Q&A session with the presenters.
Thank you all for joining us today. I'm Erin Powery, a research imaging specialist at Olympus America, and today I'm going to talk about the VS200, our research slide scanner. The VS200 was designed to address specific needs of the modern research landscape. So I'm going to give an introduction to the system and its capabilities, and after that, Dr. Michael Bienkowski will give a presentation on how slide scanning has been a part of his research. So before I talk about where we are, I'm going to talk a little bit about where we're coming from, both from the technology side as well as the applications side. The technology of slide scanning has evolved significantly over the past few decades. Traditional microscopes could take single fields of view, and researchers would hand align images and stitch them together post-acquisition if they wanted to see larger fields of view. The first generation of commercial slide scanners arose from the need to digitally archive slides. The automation of acquisition and stitching was a tremendous leap of convenience. Of course, it was just the beginning. The newest generation of slide scanners provide integrated imaging solutions that enable high throughput, automated scanning using a variety of different imaging modalities. So what does this look like in terms of the data produced? A single field of view on a traditional microscope offers a picture of what's happening in a region of interest. The higher the resolution you want to see, the smaller the area you can image. Early slide scanners stitched together larger regions of interest, enabling higher resolution views of whole tissue. And modern slide scanners are capable of high throughput whole slide digitization. So not only can we image entire slides, but we can do it using a variety of different observation methods, and we can do it really quickly. In the past couple of decades, we've seen both the technology and the applications of slide scanning grow beyond the initial designs, which were largely focused on pathology and medical applications. Slide scanning is now a formidable primary research tool. It facilitates the processing of large amounts of data in a very reasonable time. Whole slides can be imaged in a matter of minutes, even at high mag. Importantly, much of this is hands-off time, in which the scanner is doing all the work while the researchers are free to work on other things. This also means being able to image through, say, 75 brain slices changes from a huge project into something that can run overnight while you're sleeping. Not only does this save time, but it dramatically alters the kind of research goals that individual labs can work towards. Now, slide scanning also allows for the digital preservation of slide data. No longer is a whole data set represented by a few representative images, but all the data can be preserved. From a very practical standpoint, this means that slides no longer must be physically stored for long periods of time. Information is no longer lost if a slide is damaged or disposed of. It also facilitates the sharing of data across distances, whether for remote diagnosis or for long distance or more recently socially distanced collaborations. Digital preservation of slide data removes the limitations of having to be physically near the slide in order to work on the data. And modern slide scanning technology has now progressed to the point where we can image whole sections at high magnification. When you combine this with computer advances that allow a single image to be zoomed in or out, you can view a small section at high mag, like we see here, and you can zoom out progressively to lower magnification to see global localization patterns and expression levels, giving a true macro to micro imaging solution. Now, Olympus has been making slide scanners since the introduction of the dot slide in 2005. Last year, we released the VS200 as the successor to the VS120 And we worked really hard to have a system address the needs of modern research. So today, I'm going to tell you a little bit about the VS200 slide scanner and how Olympus designed a system that really prioritizes both efficiency and ease of use without sacrificing image quality. So what are some of the notable features of the VS200? We've implemented a more efficient light path in combination with our newest objectives to provide image quality while changing the body of the instrument to ensure that even when you're imaging at the highest magnifications, you get images with minimal stitching and shading artifacts. We designed the system to be very flexible, easy to upgrade, and they can handle a number of different research applications. We have 5 different observation methods. We could use either conventional or silicon oil for high numerical aperture imaging, and we can easily adapt the system to support multiple slide formats, ranging from 1x3 slides to 4x5 glass plates. We also made it really, really easy to use. So let's see the system. The VS200 comes in 2 formats, a 6-slide manual loader as seen on the left, and a multi-tray loader as seen on the right. The loader is fully automatic and can hold up to 210 1x3 slides. Now, one of my favorite features of the VS200 is that it is fully field upgradable. You can purchase a 6-slide bright field system and subsequently add fluorescence capabilities, an oiler, or a loader all in the field without having to send the system back. This allows researchers to have a system that grows along with their imaging needs. Last year, we launched our X-line objectives. Now, Olympus created a new way to manufacture the lenses that results in superior image flatness across the field of view and an increased numerical aperture at higher magnifications. This allows for greater resolution at high mag, and it also directly impacts the fidelity of shading and stitching while doing slide scanning. The use of these objectives ultimately produces a cleaner image with nearly undetectable stitch lines. Now, I mentioned the oil immersion capabilities of the VS200. Every system is capable of having an automatic oiler attached. This can be equipped with either conventional immersion oil or silicone oil. The system will dispense a preset amount of oil onto a slide and image. If more oil is needed, the system automatically detects this, stops scanning, and adds oil appropriately. We have also implemented a safety system where the scanner will not allow you to scan slides with oil on them until you've told the software that the oil has been removed. Now, the question I get every single time is, will it also clean the oil off for you? And the answer is sadly no. You have to do that yourself. However, the software does make it easy for you by moving the objective into an easily accessible position. This automation makes high magnification scanning easy and accessible with a significantly decreased chance of damaging optics due to excessive oil use. Now, you can see here the process of loading both the 6-slide and the loader versions of the VS200. Whether the trays are manually loaded into the front of the system or by a robotic arm with the loader, the thing I want to point out is that the slides never move from the trays they are inserted with. This essentially eliminates the dreaded slide dropping. We also implemented a way to interrupt a current scan and image a priority slide. This pauses the current scan. Once the priority slide is imaged, the system will resume scanning the previous batch. To further automate the process, we integrated a barcode reader into the system. As you can imagine, manually entering the information for 210 slides is incredibly tedious. So this allows the users to even further automate the scanning process. So how does this all come together? This is an example of a common workflow. Low magnification overviews of a whole slide allow you to identify your tissue. A fast multi-channel scan can be acquired at 10x, and then regions can be selected for higher magnification detail scans at up to 100x magnification. That's incredibly powerful. So now I'm going to walk you through the workflow of using the scanner So you can see how all the features I just spoke about are implemented at the different stages of scanning. So first things first, you need to load the samples. The VS200 supports various slide formats. You can see 3 of the trays here on the right. They're all RFID tagged, so the system will know what kind of tray you loaded. If you have the loader, not only will it know what kind of tray you loaded, but it can also detect whether or not a slide is loaded into a particular position, so you're not wasting time scanning slides that aren't there. The system can handle up to a 4x5-inch glass plate, so you can handle a very wide range of samples, including this glass slide here of a section of human brain. When you open up the software, you'll see a wizard that allows you to select your desired imaging modality. You have bright field and fluorescent modes. Special mode is for polarization, phase, and dark field imaging. Quick mode lets the software determine where the sample is, the optimal focus and exposure times, and expert mode allows you, the user, to directly control these things. So regardless of whether you choose quick or expert mode, the software will acquire a focus map of your sample. In an ideal world, all tissue samples would be perfectly flat and you could scan a single plane and get a beautiful image all the way across from a single focus point. We all know that real life doesn't follow those rules and many samples have topography to them. To make sure that we're able to get an in-focus image across the entire slide, our software has a sample detection algorithm and a fully automated focus map system. This places a specified density of focus points over the sample and will scan those first. It will use the Z information obtained to create a focus map and extrapolate between those points to determine the optimal focus across the whole sample. But sometimes we want all of the Z information. The VS200 is capable of imaging up to 31 Z planes at a range of up to 100 microns. This can be done for every observation method. On the right, you can see a Z stack being played through on a loop in a movie, where the slide has been imaged through 10 slices. Below that is an example of an extended focus image, or EFI, in which the Z information has been compressed into a single plane, in which the most in-focus pixels are selected and used to construct a single plane image. I'll talk a little bit more about this on the next slide. So why do we use an EFI? As you can see on the top, when your tissue is very three-dimensional, you can have in and out of focus areas even within a single field of view. For an EFI, the VS200 will acquire a Z stack and then determine the most in-focus pixel and assemble those into a single image. This has 2 benefits. First, you don't have to look through different planes to see all the information. And second, it dramatically decreases file size, which for those of you who are experienced with slide scanning and Z stacks, or the combination of Z stacks and slide scanning, know that the file sizes can end up being exceptionally large. This is also something that's very useful when you're imaging multi-channel fluorescence and thicker brain slices, where the labels in different channels could be in completely different planes, as well as having a certain amount of three-dimensionality to the tissue. The EFI provides a clear picture of everything that's labeled within the tissue in a file that's the same size as a single plane image. But what else can you do with the Z data? Here we see the EFI again on the page in the upper left. We can also do a max projection where we're choosing the brightest pixels for our projection, or we can apply various deconvolution algorithms ranging from 2D decon options like a Wiener filter to true 3D deconvolution using an advanced maximum likelihood algorithm. So we're able to provide a whole host of image processing options to allow you to select the best image output. So now that we've seen the hardware and software capabilities, I'm going to touch on the applications. So what do people use slide scanning for? Well, the short answer is a whole lot. And we wanted to make a system that addressed as many of those needs as possible. The VS200 can image up to 5 different observation methods. The base system comes with bright field and dark field observation methods. Dark field is particularly useful for finding unstained samples that will be subsequently imaged in fluorescence without unnecessarily photobleaching your sample. Fluorescence is an add-on. We can accommodate up to 8 different filter cubes in a mirror turret with options for high speed, high accuracy imaging with fast switchable LED light sources and filter wheels. We have the ability to do phase contrast imaging, which is particularly useful for people imaging monolayers of cells. And last but certainly not least, we can do simple polarized light imaging, which allows for the visualization of birefringent structures such as amyloid plaques. Having all of these capabilities in a single system provides a powerful research instrument that can easily be adapted as experimental needs change. Here's a list of some of the research that's commonly being done with slide scanners every day. And if I'm being honest, researchers are coming up with new ways to utilize these machines all the time. Right now, I'm going to take a minute to expand on the last item on this list, multiplexing, because it's a very powerful tool that's becoming more and more prevalent on the research landscape. So we implemented onboard software specifically for use with aligning multiplex images. If you're not familiar with multiplexing, it's a methodology for labeling multiple different epitopes within a single sample. Samples can be labeled with up to 5 dyes and imaged. The slide can then be stripped and reprobed with different antibodies. Our software uses a single channel, which is usually DAPI, as a guide for aligning, allowing you to construct many different multi-channel images into a single image. Here's an example of a 5-channel slide acquired at 20x using a commercially available multiplexing panel from UltaView. You can see the multi-channel image being constructed as I add back in channels. Now, this process can be repeated with another panel and the layers toggled in any combination you need. This is not prohibitive from a time perspective, as the average scan time of these samples are between 10 to 15 minutes per slide for 5-channel fluorescence. So multiplexing is proving to be an extraordinarily powerful tool in modern biology, and the VS200 was very specifically designed to deal with multiplexing samples. So last but certainly not least is the topic of data handling. What on earth do you do with all these scans? Our native file format is the VSI, and many widely used analysis programs such as Halo, Visiopharm, and Imaris natively read our file format. But to make sure that file format's never an issue for analysis, we can convert or directly save all images in a variety of standard formats so you can analyze your images on the platform of your choice. Olympus does have their own suite of analysis options. VS Detect is our standard threshold-based counting module. It's straightforward, it's easy to use, and you can export the results directly to Excel. But where it really shines is when it's used in combination with our new AI module. True AI is our highly flexible deep learning artificial intelligence analysis module. Essentially, it's a toolbox for customized image analysis. You can train the software to detect complex objects. This works for any observation method. For fluorescence, it can take into account the localization and morphology of multiple channels. When you train the software to detect your object of interest, you can use the counting module then to count these objects and it's not threshold based. These objects that you count can then be used as a mask to facilitate traditional counting methods within these objects. So like as in a parent to child relationship. So for example, if you wanted to identify how many nuclei are located within these pancreatic islets, you could use AI to determine the islets and then use that signal to make a mask and then do a traditional thresholding based DAPI count to identify how many nuclei are present within these. It's really a very powerful tool. Ultimately, true AI is more accurate and reliable than traditional methods of counting. It works well on bright field images as well as fluorescence. It's a highly flexible option that opens up doors for image analysis that were previously very challenging. One last thing I want to touch on that has become extremely important this year is data management for remote viewing. We have our Net Image Server, which is a database software that integrates seamlessly with the VS acquisition software. Within this, you can set up individual permissions to access images, and it comes with a web-based viewing platform. So regardless of your operating system, you can remotely access your data, view, annotate, share, and screenshot your data remotely. So people working from home or collaborating long distance, this becomes a very powerful tool for being able to securely access your images online. In addition to this, we offer Olivia, which is our free image viewer. Olivia gives any user the ability to open up the files, take basic measurements, save them, extract layers, crop, and it's a very handy free tool that allows anyone to do basic image manipulation without having to go to a specific analysis station. So I hope this has been really informative. And in summary, the VS200 is a research slide scanner that offers exceptional image quality on a platform that's easy to use, flexible, and easily upgradable. Thank you for your time, and we'll be happy to answer any questions you have.
Thank you, Erin, for presenting on the VS200 slide scanner. If there are any questions, you may post them in the questions pane of the webinar control panel. I will now turn it over to Dr. Michael Bienkowski.
Thank you everyone for coming today, and thank you to Olympus for inviting me to highlight my research program. My name is Michael Bienkowski, and I'm an assistant professor of physiology and neuroscience in the Stevens Neuroimaging and Informatics Institute and Zilkha Neurogenetic Institute at the University of Southern California. My research is focused on understanding the multi-scale organization. Thank you everyone for coming today, and thank you to Olympus for inviting me to highlight my research program. My name is Michael Bienkowski, and I'm an assistant professor of physiology and neuroscience in the Stevens Neuroimaging and Informatics Institute and Zilkha Neurogenetic Institute at the University of Southern California. My research is focused on understanding the multi-scale organization of the hippocampus. That is, how are different types of information represented across the entire hippocampal structure? The hippocampus is one of the most well-studied regions of the brain that is affected by a variety of neurological diseases like Alzheimer's and epilepsy. From the gross anatomy of the human hippocampus, we can see it has a truly unique and distinctive architecture. Although some often treat this brain region as if it's one singular structure, you can see here that there's a great deal of variation across its curved longitudinal axis. Its posterior pole is thin and curled, whereas the anterior end is thick and has several noticeable ridges along its surface. In general, this distinct structure is largely conserved across all mammals. In the human brain, the hippocampus is positioned within the ventral temporal lobe, with its long axis oriented in the anterior-posterior direction. Here I've colored the major hippocampal subregions of the dentate gyrus, CA regions, and subiculum to note their anatomical positioning. As we go backwards through evolution, this axis is shifted, and so the orientation of the hippocampus in rodents is that the long axis is dorsal ventral. With our understanding of this rotation of the hippocampal axis, the mouse and human hippocampus are believed to be both structurally and functionally homologous despite a considerable difference in size. Here again is another example of the relationship of the hippocampus in rodents, monkeys, and humans, showing the similarity of this long, curved axis and its positioning in the brain relative to the entorhinal cortex and the cytoarchitectural organization. Now what you may notice in these diagrams is that there's corresponding terms of ventral, intermediate, and dorsal, as well as anterior, body, and posterior in the monkey and human, are labeled in these diagrams, but there are no defined boundaries which separate these regions. And as you move along this axis, no one has really known exactly where one part stops and another part begins. In fact, it's not even entirely clear how many parts of the hippocampus really exist. The question of how this hippocampus is organized as a whole structure has generated three somewhat incompatible answers from three separate fields of neuroscience. The behavioral neuroscientists have proposed a dorsal-ventral dichotomy, Anatomists have focused on a tripartite gradient, and gene expression and transcriptomic studies have identified multiple subregions or gradients across the hippocampus. And the goal of my research is to try to bridge the gaps between these fields of neuroscience, to try to understand how to synthesize these different modalities and understand multi-scale organization in the hippocampus. And the work I'm going to talk about today began by investigating the gene expression and anatomical connectivity within the mouse. To understand the organization of gene expression across the whole hippocampus, I analyzed in situ hybridization data from Allen Institute's website and mapped their spatial distribution to create the Hippocampus Gene Expression Atlas, which identified 20 new molecular domains across the dentate gyrus, CA3, CA2, CA1, and subiculum. Once I had a complete atlas of genetically defined hippocampal subregions, I delineated their input and output connectivity using multiple combinations of anterograde and retrograde tracers as part of the mouse connectome project at USC. The foundation of this project is the double co-injection approach, where we inject two pairs of anterograde and retrograde tracers to simultaneously visualize the inputs and outputs of two brain regions in the same brain on a Nissl background. By using both anterograde and retrograde tracers, we can cross-validate many of our results by labeling the same pathway with two different types of tracers. In addition, we can perform triple anterograde tracing to visualize 3 axonal pathways, or quadruple retrograde tracing to visualize the distribution of projection neurons. All of this data is then imaged using whole slide scanning microscopy so that we can analyze the connectivity patterns across all levels of the hippocampus. In total, this project contains data from 79 mice and over 177 tracer injections, which you can access all of this image data online using the iConnectome viewer along with other visualization tools at the Mouse Connectome Project website. The boundaries of the 20 molecular subregions of the hippocampus and subiculum were mapped onto the stereotaxic Allen Reference Atlas and aligned into a 3D model of the hippocampus structure. Here you can see how the boundaries of these regions shift as you move from rostral to caudal levels. By injecting anterograde and retrograde tracers into each of the 20 molecular domains, I was able to assemble a comprehensive hippocampus connectome wiring diagram that includes the brain-wide inputs and outputs of all 20 hippocampal regions. Overall, we found that gene expression patterns were highly related to anatomical connectivity patterns. For example, at this caudal level of the subiculum, the red and yellow regions of the atlas on the left correspond to the neurotensin and TLE4 gene expression patterns shown here on the right. If we then look at connectivity patterns at this level, we see that anterograde and retrograde tracer labeling produced by injections into downstream targets of the subiculum reveal anatomical connectivity patterns that are highly related to the boundaries defined in the atlas image. Using our quadruple retrograde tracing approach, we were able to retrogradely label almost every neuron at this level to produce an image of anatomical organization of projection neurons that's highly similar to the gene expression atlas on the left. More recently, we have begun analyzing Allen Institute's human in situ hybridization data and have found evidence that the human subiculum also contains similar gene expression layers. Here you can see that the gene expression patterns for 3 genes, the serotonin receptor 2A, neurotensin, and the muscarinic cholinergic receptor 2, alongside with adjacent Nissl sections and accompanying atlas section. In the subiculum, we can see that there's an absence of HTR2A, whereas neurotensin staining cells are in the superficial area, and the CHRM2 cells are located deep near the alveus white matter. In prosubiculum, we can see a lot of HTR2A cells, an absence of neurotensin staining, and a thin, deep layer of CHRM2 cells as well. Together, these 3 complementary gene expression patterns show that we can define boundaries in the human hippocampus in much the same way as in the mouse. While not every single cell gene expression profile is identical, we can see that the mouse dorsal subiculum and posterior human subiculum are not only structurally and functionally homologous, they also contain conserved cell type architecture and gene expression. In both mouse and human, we can see that NTS and CHRM2 cells are defined both by their anatomical location relative to the molecular layer and alveus, although these positions are flipped due to the rotation of the longitudinal axis across species. As part of a pilot project with the USC ADRC, our lab is now using slide scanning microscopy to investigate how Alzheimer's disease pathology is related to human gene expression patterns and their unique cell types. The Gallius stained images on the left and the 4G8 stained images on the right are two of the primary stains for post-mortem evaluation of the tau neurofibrillary tangles and amyloid plaques, the two major hallmark signs of Alzheimer's disease. By scanning these sections, we can both examine the broad distribution of the pathology across the hippocampus and also zoom into these images and observe the pathology at a cellular resolution level. Ultimately, our goal is to use gene expression atlases to link Alzheimer's pathology to high-resolution diffusion tractography to help us understand which cell types are susceptible to Alzheimer's disease and how Alzheimer's affects their neuronal connectivity. In addition, our lab is using 3D microscopy in combination with clarity and shield tissue clearing approaches to image large volumes of human postmortem tissue, which can then be registered to MRI datasets. The image here shows an 8 mm thick sample of human hippocampus along with the parahippocampal and fusiform gyrus, and using 2-photon imaging and immunohistochemical labeling, we can reveal neuronal connections in Alzheimer's disease. So in summary, our research has shown that the hippocampus can be divided into as many as 20 molecular domains with a high degree of similarity between the gene expression and anatomical connectivity patterns. Our initial investigations into human hippocampus suggest that there is a homologous organization of gene expression, and this adds to our understanding of structural and functional homology between the mouse and human. Finally, we believe that developing a human hippocampus gene expression atlas will provide a critical bridge to understand the molecular underpinnings of Alzheimer's disease and help guide the translation of mouse research to human clinical development. Finally, in closing, I want to acknowledge the members of my new lab at USC, as well as the many members of the Mouse Connectome project team that helped to generate and analyze these data. In addition, I want to thank our collaborators at USC and in the Brain Initiative Cell Census Network, as well as our funding sources who have helped to support this work. And finally, thank you again to Olympus for the opportunity to present this research.
Thank you, Dr. Bienkowski, for your presentation. Also, thank you to Erin Powery for your presentation. I will now answer or ask questions that we have received during the talk. The first question, what detector and light sources are used for fluorescent imaging?
Yes, this is Alec here. I'm the product manager for the VS. We actually have 3 different camera options that you can choose. A simple CMOS fluorescent camera that we produce ourselves, as well as using the Hamamatsu Flash V3 SCMOS camera and Flash Fusion SCMOS cameras. For light sources, we can provide a broadband white light Xylus from Excelitas, or a selectable channel light source from Turbo, also from Excelitas.
Thank you, Alec. And the next question, how does multi-observation scanning work?
So basically, what happens is you run your normal scans. Let's say you pick bright field, dark field as your observation methods that you want to do, and you run them sequentially. So essentially, you would scan once with the bright field, complete that scan, and then at the very end, before you save the file, you just go ahead and hit add a scan, and you select a whole new project and you run it again. This will add a new layer to the file, allowing you to add multiple observation methods to the same file type on the same slide in the system without having to change anything or make any more adjustments.
Great. Those were the only questions that we received today. Again, thank you to Erin, Michael, and Alec for your presentations and answers to the questions on today's webinar. I hope that everyone enjoyed the talk. This presentation will be available on demand starting tomorrow on the neuroscienceweek2020.com page. Today's Neuroscience Week events also include a VS200 demonstration at 1 p.m. Eastern Time and a special session on single-cell manipulations with a nanosyringe presented by Cytosurge at 3 p.m. Eastern Time. Be sure to visit www.neuroscienceweek2020.com to see what else we have in store for the week and to enter into our coloring contest. Thank you for your time.